Hello, thank you for the invitation. My name is Di Francesco. I'm a neurologist working in Italy, San Gerardo in Monza, and here I present the collaborative work of a single case, a single patient, followed by different institutions here in Italy. And we describe our experience with in this patient with KCNT1-related drug-resistant focal epilepsy, who was treated with she is treated with the antidepressant drug fluoxetine as a precision medicine approach. I have no disclosure. So briefly, Noemi is a girl. Now she's 20 years old. She was born after an uneventful, a normal pregnancy with a normal delivery and with a normal psychomotor development during the first 3 years of life. She doesn't have any history of epilepsy or other neurological disease. And at the age of 3 years, she started with focal seizure with a temporal semiology that rapidly became very severe and frequent, like daily seizures. In the first 16 years of her life, she was followed in a different area of our country, but not in our institutions. And EEG reported the left temporal epileptic spikes, and brain MRI was normal. Then she came to the Neurological Institute Carlo Besta of Milano at the age of 16, reporting daily focal seizure, as I already described, with also severe intellectual disability and also anxiety disorder and aggressiveness. Until that time, she was treated with different anti-seizure medication, including lamotrigine, oxcarbazepine, and clonazepam, and other compounds that all resulted in ineffective. At the Institute Besta, this patient repeated brain MRI that was normal and CGH array that was unrevealing. On the right side of the screen, you can see her EEG that showing in the red squares epileptic spikes on bitemporal regions and a poor background organization. Then the patient underwent to our NGS target panel for the screening of the epileptic genes for epilepsy, discovering a novel heterozygous missense variant in KCNT1, leading to a substitution on the residue 937 that resulted de novo in the proband with parents wild type. This variant is localized here in the RCK2 domain, which is important for the correct function of the channel. And as you can see on figure B, this residue is highly conserved among different species. So together with our colleagues of different structures, we decided to functionally characterize the properties of this novel variant using a CHO model, including the expression of both wild type and mutant channels, including both homomeric and heteromeric conditions. The experiments, as you can see on the right side, showed a significant, strong gain of function effect, as expected with the other KCNT1 variants, both in the homozygous and in heterozygous condition determined by this novel variant. So, in order to identify a possibility of precision medicine approach, we looked for considering the gain of function effect to KCNT1 blockers. So starting with our previous experience, we tested in vitro, our colleagues tested in vitro the effect of quinidine, a known old antiarrhythmic drug that has been already tested in a few KCNT1 patients, however, with contrasting clinical results, also considering the severe concern about the cardiac toxicity However, this compound resulted a good channel blocker because could block also the KCNT1 wild type form and the mutant form of the channels. However, we recently demonstrated the effect of the antidepressant drug fluoxetine that is able to reduce, to block efficiently the effect, the activity of potassium channels. We tested in vitro also this drug that resulted also efficient to reduce again the wild type form and both the mutant form of this channel. So, considering the good tolerability of this drug, general good tolerability that is widely prescribed, the presence of frequent focal seizure not responding to several anti-seizure medication, and these in vitro data that I already showed you, we started at the age of 18 years old. The patient started with a precision approach with fluoxetine, considering the in vitro data. She started with an initial dose of 10 milligram per day that was very slowly increased until 60 milligram per day, until 1.2 milligram per kilos, reaching a plasma level at the plateau of more than 400 nanogram milliliters, corresponding to the what is used for the antidepressant chronic treatment. And these are the results. The actual situation now, she is older than 20 years old. This drug is well tolerated, and we observe the complete disappearance of seizures with only sporadic episodes that occur during COVID-19 infection. She is under treatment until now, after more than 2 years since the beginning, and acetazolamide was suspended. That was used during the clusters of seizures. At the moment, she did, and the patient and the family didn't want to reduce the other anti-seizure medication, but is now working on this. And also we can report a significant improvement of both behavior and mood, considering that the patient could achieve a high school graduation. This is briefly the chronological order of her pharmacological treatment since the onset of seizure at 3 years old until now, with the introduction at 18 years old of fluoxetine. And this is now the EEG trace during treatment that was last year, 19 years old, with a better organization of the background and no epileptic discharges. This is all the authors that contributed to this work that is now under review into epilepsy. Thank you very much.